Hi, everyone, and thank you for joining us today. Welcome to today's Microscopy Masters webinar, focusing on investigating immunomodulatory antibodies for cancer therapy. Before the webinar begins, we'd like to go over some quick house rules for the webinar. I'm sure you've all noticed by now, but you do have a webinar navigation panel with different sections. If you'd like to see more clearly one specific section, you can click the expand icon, and that's located in the top right corner of each section. Today's webinar will last about 45 minutes, leaving approximately 15 minutes for Q&A. So you are welcome to submit any questions that you have throughout the webinar via the chat section located within your panel. Sonya will be answering your questions live during the Q&A session. And if we're unable to get to your questions, then we will email them over to Sonya to address them individually. Within your panel section, you'll also find a handout section. Here you can find our company brochure telling you a little bit more about ONI and our offering, so please feel free to have a look there. And please note that today's webinar is being recorded. And if you'd like a copy of this webinar, please feel free to send us an email afterwards, but it will be stored on the resources section of our website. And now that we've gone over the house rules, I'd like to introduce Dr. Sonya James. Sonya did her PhD at the MRC Clinical Sciences Center at Hammersmith Hospital, looking at sites of transcription in cell nucleus, where she utilized confocal and electron microscopy, as well as a fledgling super resolution technique, SMI, in collaboration with the Kirchhoff Institute for Physics at the University of Heidelberg. After working as a postdoc in the same lab, Sonya moved to a position as a specialist microscopy technician in the School of Cancer Sciences at the University of Southampton. In this post, Sonya can indulge her enthusiasm for all manner of microscopy techniques, from simple bright field and fluorescence imaging to higher-end super resolution and electron microscopy. Her role has recently been merged with the University of Southampton's Biomedical Imaging Unit, where she's looking forward to trying her hand at some spatial biology techniques and bringing the possibilities of super resolution to a wider audience. And with that, I will now pass the presentation over to you, Sonya, to begin.
Okay, thank you very much, Carolyn. Ooh. Sorry, have you got, does that look okay? Because my video has just come up.
Yep, you're perfect, Sonya.
Okay.
We see your first slide.
Okay, so I work at the Center for Cancer Immunology. It's part of the School of Cancer Sciences in the Faculty of Medicine at the University of Southampton. The Center for Cancer Immunology was opened in 2018. And it's the first centre in the UK which is dedicated to cancer immunology. The work I'm going to present today is mostly work carried out by a research fellow in our group, Ben Yu. And it encompasses mostly work that's published in these two papers. Firstly, Nature Communications, which was published in 2021, and then a Nature paper from earlier this year. So, in recent years, cancer immunotherapy has revolutionized cancer treatment because it can significantly improve the survival of patients in late-stage cancer. So currently, mainstream immunotherapies include antibody therapy and cell therapy, but antibody therapy accounts for the majority of treatments in the clinic due to its efficacy in a broader range of cancer types. Also, antibodies are very versatile. They can be generated to target any protein. They can be engineered to tune their properties, and they can be produced at scale in vitro. So they're very attractive as therapeutics. So there are three main types of antibodies that are used for cancer therapy. We have direct targeting antibodies, checkpoint blockade antibodies and the immunostimulatory antibodies. The direct targeting antibodies bind to antigens which are present on the tumour cells themselves, and they mediate cellular cytotoxicity. Well-known examples of this are rituximab, which binds to CD20, and it's used to treat many types of cancer and also autoimmune disease. And also Herceptin, which binds to HER2, and it's used to treat, commonly used to treat breast cancer. So in contrast to the direct targeting antibodies, we have the checkpoint blockers and the immunostimulatory antibodies. And these are what are called immunomodulatory antibodies. So in contrast to the direct targeting antibodies, they bind to immune cells and regulate their function. So those that bind to a receptor and impede the receptor's interaction with its usual binding partner or ligand are what are called the blocking antibodies. And then by contrast, the immunostimulatory antibodies are what we call agonists, so meaning that they can function as synthetic ligands to activate a receptor. So the aim of using these immunomodulatory antibodies as therapy is to boost anti-cancer immune responses. So the tumour microenvironment is often immune suppressed. So development of therapies that can boost the immune system into action against tumour cells are key to successful cancer treatment. So the action of immunomodulatory antibodies is largely T-cell mediated, and it offers the promise of anti-cancer immunity. So once you've stopped using the drug, you should still have immunity to that cancer. So the work I'm presenting today is going to be mainly focused on the immunostimulatory antibodies. These are the ones that act as an agonist. That is, they can bind to receptors and activate them in the same way that the receptor's endogenous ligand would. There are multiple targets that could be used for possible therapeutic antibodies. Many are from the family of receptors known as the tumour necrosis factor receptors, or TNFRs. So TNFRs are important regulators of the immune system. They mediate proliferation, survival, differentiation, and function of immune cells, which makes them very attractive targets for immunotherapy. So much of the work presented today focuses on one such TNFR, CD40. This is a surface molecule expressed on both immune and non-immune cells, and CD40 activation is crucial for an effective immune response. So during immune activation, in response to a pathogen or a toxic molecule, an antigen-presenting cell, or APC, presents the foreign antigen in a complex with MHC class II to a CD4 positive T cell via the T cell receptor. The CD40 ligand then present on the CD4 T cell in turn engages the CD40 receptor present on the APC. This produces a signaling cascade from the receptor, which ultimately licenses the APC to prime a CD8 T cell response. The primed CD8 T cells undergo massive expansion and differentiation into cytotoxic effector cells, which then work to clear the body of the offending infection. So in this way, the immune system should be able to eliminate tumour cells from the body. However, this is not always the case because the tumour microenvironment is often immunosuppressive. It has high expression of inhibitory receptors and ligands and a lack of ligands for the stimulatory receptors. So the cancer cells are often not recognised as targets. So in the absence of a CD4 T cell activation, an agonistic anti-CD40 antibody could be used to activate the CD40 receptor, and in this way induce signaling cascade leading to immune activation. So many anti-CD40 antibodies and other TNFR antibodies have been investigated, and some are being used in clinical trials. Not surprisingly, the level of agonism, that is how good the antibody is at activating the receptor, correlates with their efficacy. And a lack of agonism is associated with clinical failure, but also excessive agonism produced toxicity-related complications. So it's clear that a better understanding of the molecular features that underlie differential TNFR agonism is critical for the generation of agonists with the desired level of activity we need to elicit therapeutic benefit. So the work of the antibody and vaccine group at the CCI has been focused on investigating the key determinants that underlie antibody efficacy in eliciting a beneficial immune response. So these include the involvement of Fc gamma receptors and also elements of the antibody itself, such as the antibody epitope, binding affinity, and isotype. The work I'm presenting today focuses on these last three determinants. Antibody epitope, so antibodies to different epitopes produce differential agonistic activity, which is related to differences in receptor clustering. Similarly, the binding affinity, reducing affinity actually boosts the agonism. And again, this is mediated through differences in receptor clustering. And then the antibody isotype, so switching the isotype of CD40 antibody can convert it from an antagonist to an agonist. So I mentioned that receptor activation was mediated by receptor clustering. TNFR activation follows this paradigm of ligand-induced receptor oligomerization, which is a general characteristic of membrane receptors. So agonistic antibodies also elicit this oligomerization or clustering, with distinct levels of agonism leading to distinct forms of receptor clustering. This was shown in Ben Yu's 2021 Nature Communications paper, where he studied the agonism of anti-CD40 antibodies with different epitopes. So Ben carried out epitope mapping by various methods to deduce where the epitopes of a panel of CD40 antibodies were. And these antibodies encompassed both agonists and antagonists, and they included some antibodies which are currently in clinical trials. So he found that CD40 agonists induce differential clustering. So in this experiment, we have Jurkat cells which express human CD40, shown here in green, and they were assessed with this panel of clinically relevant CD40 agonists and antagonists. So in the top two panels here, we have confocal Z-stack projections showing that there are distinct patterns of receptor clustering depending on the treating antibody. And the bottom panel shows histograms for the upregulation of CD23 on human transgenic B cells, and this is a measure of agonistic activity. So on the left here, we can see that in the control, untreated control, we have no clusters, no CD23 expression, and with the CD40 ligand, we have these small distinct clusters and a high upregulation of CD23. And then with the panel of different epitopes, we can see that there is a range of different degrees of clustering. We also can see that by switching the isotype from the human IgG-H1 to human IgG-H2, can change an antibody from an antagonist to an agonist. So just in this panel here, we have an antagonistic antibody, 24.2.1, and it shows no clustering and no agonistic activity with either isotype. And the agonistic antibody CP shows these small distinct clusters with both isotypes, and also the upregulation of CD23 with both isotypes. Whereas the Chi-Hub 74 and 341G2 don't show any clusters or agonistic activity with the H1 isotype, but they do with H2. So interestingly, also this superagonist, this 341G2, which is the most agonistic anti-CD40 antibody, develops these clusters that are this rod-shaped, and this was not observed in any other with any of the other agonists investigated across all the different TNFRs. So to confirm these observations in more physiologically relevant cell populations, Ben used CD40 expressing immune cells and treated with the anti-CD40 antibodies or the CD40 ligand. And consistent with the previous data, the same differential receptor clustering was recapitulated in normal human B cells isolated from healthy donors, B cells isolated from human CD40 transgenic mice, and also human monocyte-derived dendritic cells. He also looked at different TNFRs, so he did the same experiments with two other TNFR antibodies, 41BB and OX40. And the same different pattern of differential receptor clustering was also seen with both of these. So indicating that it may be a conserved feature among the TNFR family. And similar to CD40, the human IgG2 isotype conferred more prominent clustering and also stimulated higher levels of NF-kappa B here, which is also a measure of agonistic activity. And the same was true for OX40. So if we go back to this initial figure, we were expecting that the size of the receptor cluster and or the degree of receptor clustering would positively correlate with agonistic activity, with the strongest agonist displaying the largest and or most extensive clusters. However, as you can see, this is not the case. So CD40 agonists displaying strong agonism, such as the CP and the CD40 ligand, induce notably smaller clusters than weaker agonists, such as Chilob74. So to investigate the receptor clustering features underpinning this agonistic activity in more detail, we decided to employ the super resolution microscopy technique of D-storm. So I'm sure most of you know how D-storm works already, but I'm just going to do a very brief and simple explanation just in case anyone isn't sure. There are many excellent resources on the internet and on the ONI website. So if anyone would like to learn more about how D-storm works and how to set up D-storm experiments, they can have a look there. So the reason that we need super resolution is that the lateral resolution limit of the conventional light microscope is only about 200 nanometers. And in Z, the Z resolution is less, about 400 to 500 nanometers. So this limit occurs because, as we know, light travels in waves, and the emission distribution of an object is dependent on the wavelength of the illumination system. So in theory, this means that with the light microscope, 2 points must be greater than 200 nanometers apart for them to be detected as two distinct points. Any closer than 200 nanometers, and they'll no longer be able to be discerned as two distinct points. So this means that on a conventional light microscope, if we take an object which is less than 200 nanometers in size, such as, for example, this ring-like structure, we just see the object as a large blob and are unable to discern that it is in fact a ring structure. If we could have the particles of the structure emitting light one at a time, it would help to discern the underlying structure. And this is the principle of D-storm microscopy. So certain fluorophores, when they're excited by a high power laser, will switch on to the on state and then immediately switch back off into their dark state. And we call this switch between off and on and back to off again, blinking. So if we image a sample under high laser intensity, the fluorescent molecules will start to blink, but crucially, they will not all blink in unison. And since the light detected from each blink is coming from one molecule, we can use some nifty physics here to calculate the probability of the localization of the molecule from its emission distribution. So for our ring-like structure example, if we induce fluorophore blinking and collect sequential images, if we collect enough images, we should have a record of a blink from each of the particles in the object. So each blink allows us to calculate localization probability. So we can start to build up a picture of the underlying structure. And the precision with which we can locate the location probability, i.e. how accurate we are, is dependent on a number of factors, including the point spread function of the microscope, the number of photons collected, the pixel size, and the background. But typically this can get down to about 20 nanometers or less, i.e. we can, the molecule is within 10 to 20 nanometers of where we say it is. So if our sampling is good and we take enough frames, so typically we need to take more than about 10,000 or more, we should have a record of a localization from every labeled molecule in our sample. And we can use this data to produce a localization map or an image of our sample, which has a resolution of below 20 nanometers rather than greater than 200 for wide field imaging. So if we go back to our CD40 data, we used Jurkat cells expressing CD40 and treated them with Alexaflor 647 labeled anti-CD40 antibodies, and then checked for receptor clustering. So the images shown here are from the cell surface close to the cover slip. So they're taken in turf mode on the nanoimager to increase the signal to noise ratio. So the top panel shows confocal images where we can see the receptor clustering. And we can see the rod-like clusters with the 341G2 antibody. The bottom panels here show the D-storm data, where each dot in the zoomed image represents a single molecule localization. So we did some clustering analysis of the D-storm localizations using HDB scan, and this revealed that receptor clusters observed under the confocal microscope were further comprised of substructures. And we can see here that the organization of these substructures differed between the different antibody epitopes. So analyzing these substructures, we find that the stronger agonist, CP, induced smaller clusters as defined by cluster area, but they had significantly higher receptor density, almost a log higher than the weak agonist, Kylog 74. Interestingly, the cluster area exhibited an inverse correlation with the receptor density for all of the antibodies, which might indicate that larger clusters may not be as efficient at recruiting more receptors as the smaller ones. The 341G2 superagonist didn't show such high density clusters. However, we know that the nature of the 341G clusters in these rod-like formations is very different to that of the other epitopes. So it may be this underlying architecture which affects receptor activation rather than subcluster density. So given the complexity of TNFR signaling, it's likely that multiple factors, including the cluster number, their size, and their density, and their superarchitecture, all have a bearing on their agonistic activity. So Ben then went on to look at antibody affinity. So during an antibody response, or antibody responses during infection and vaccination, typically undergo affinity maturation. And this leads to the selection of B cell clones that produce high affinity antibodies. And these antibodies are required for efficient pathogen opsonization and neutralization. And high affinity is crucial for the activity of naturally occurring and conventional direct targeting therapeutic antibodies. But in comparison to direct targeting antibodies, the rules governing the activity of agonistic antibodies are more complex and multifactorial, and they're determined by a combination of determinants, as we discussed, the antibody epitope, the isotype, and Fc gamma receptor requirements. So previously, no study had systematically examined the impact of affinity on the activity of the antibodies. So Ben used the crystal structure of CD40 in complex with the clinically relevant antibody KYLOB74 as a model to design affinity mutants. And what he found was that the affinity agonism relationship revealed a bell-shaped curve whereby B-cell activation increased from the parental KYLOB74 wild-type antibody with reducing affinity. So we wanted to see whether this enhanced agonism could be translated into therapeutic benefit. So he took mice treated with tumours. They were treated with the CD40 antibodies, and we can see that although the wild type antibody here in the dark circles conferred some survival benefit, the low affinity version was significantly more effective. In addition, there's evidence of strong immunological memory. So when the mice that survived the initial experiment were re-challenged with tumour, mice treated with both the wild type and the low affinity antibody previously both survived. So these data collectively demonstrate that low affinity anti-CD40 antibodies are able to mediate agonistic activity in vitro and in vivo and exhibit this potent anti-tumour activity. So these experiments I've just showed you were on with KYLOB74 on the anti-CD40 antibody, and Ben also carried out similar experiments with another TNFR receptor, 41BB, and as well as an antibody to PD1, which stimulates in inhibitory signaling in immune cells and is an important therapeutic for autoimmune disease. So in both of these cases, he saw the same results, that lowering affinity boosts the agonism. So we wanted to investigate how the affinity affected the agonism, and we know that differential clustering affects agonistic activity. So we hypothesized that low affinity antibodies, which retained sufficient antigen binding, would induce higher agonism through increased receptor clustering. And we find that this was indeed the case. So here we have some confocal images of Jurkat cells treated with the wild type antibody and with the affinity variants. So in this experiment, the cells were left undisturbed on the cover slip, and we could see clustering forming predominantly at the cell-to-cell junctions. In order to quantify the clustering, we used a clustering index, which is basically a function of the mean fluorescence intensity at the cell-to-cell junctions in relation to that at the cell periphery. So a larger clustering index denotes increased clustering. So the low affinity KYLOB74 variants induced significantly greater levels of receptor clustering. So they had a higher clustering index than the wild type, which did not induce appreciable clustering. So in order to have a better look at the clusters formed at the cell-to-cell junctions, We again employed D-storm microscopy. So here you can see at the top wide field images of cells treated with various antibodies. So we have the wild type KYLOB74 mouse 1 antibody and the low affinity variant, and also the KYLOB74 human 2 variant, which we know has increased agonism. So we can see there are significant cell-to-cell clusters formed with the human 2 variant and also the low affinity variant. And consistent with our clustering index data shown previously, we could see that the density of receptors was significantly higher at the cell-to-cell junctions with the strong agonists in comparison with the weak agonists. Importantly, it was also significantly higher than the increased density that would be expected because there are 2 membranes rather than just 1. We can see that here with the slight increase with the not treated cells. So the increased agonism correlates with receptor clustering at the cell-to-cell junctions. So we carried out some clustering analysis using HDB scan, and this was done on the CODI platform. So we wanted to investigate the substructural makeup of these cell-to-cell clusters. So here we can see a wide field image. This is then the STORM data, and this is just a zoomed in area here of the cell-to-cell junction. And we can see that these cell-to-cell clusters are in fact made up of subclusters. Analysis of these subclusters revealed that they had a greater receptor density with the more agonistic antibodies than with the weak agonist. So this agrees with our previous data suggesting that smaller, denser clusters induce greater receptor activation. So far, clinical responses for cancer treatment using agonist antibodies that target members of the tumour necrosis receptor family have been disappointing. And although multiple contributing factors might be involved, many of these approaches use high affinity antibodies. So this raises the possibility that we could improve responses through affinity optimization, because we see that with the immunomodulatory antibodies that lowering affinity can increase agonism, and this lowering affinity enhances receptor clustering, and that higher density receptor clusters enhance the agonism. So use of lower affinity antibodies may enhance clinical outcomes. So the future work that we want to look at, firstly, we want to look further into these cell-to-cell clusters. So to do that, we've started doing some analysis of double-labeled cell-to-cell clusters. So here we have a confocal 3D Z projection, and we have human CD40 GFP cells mixed with human CD40 M cherry cells, and then they're treated with the KYLOB74 human 2 antibody. And we see that we get clustering and we get this kind of cap formation at the cell-to-cell junctions. So this is what we want to further investigate. So we have started doing some 2-color D-storm with these same antibodies. We then use an anti-GFP or anti-M cherry nanobodies to boost the signal. And we find that at these cell-to-cell junctions, we get different patterns. We get intermingled clusters. We get these clusters sitting side by side. And also we seem to get what looks like flipped membranes, where this is the GFP cell on this side and this is M cherry. So it looks like the membranes have been flipped. And so what we want to look at with this is to do some 3D storm of these cell-to-cell clusters to see what's happening with these these membranes that look like they've been flipped. We'd also like to go on to do some single particle tracking to look at the formation of the cell-to-cell clusters. So we know that with some antibodies, it's very rapid. It only takes a few seconds after adding the antibody before you start to see the clusters. And with other antibodies, it's much slower. And we'll also want to look into quantification, so the actual numbers of receptors which are needed to form the greater agonism. Okay, so I'd just like to acknowledge Ben Yu, who did most of the work here in conjunction with the Antibody and Vaccine Group at the CCI. And also funding for our ONI nanoimager came from the Mark Benevolent Fund. And I'd also like to thank the guys at ONI, especially Pradeep and Frankie, for all their help. Thank you very much for your attention.
Right, wonderful. Great. So thank you so much for your presentation, Sonya. We are now going to open up the floor to questions, which Sonya will be answering live. And once again, if you do have any questions, please feel free to drop them in the chat section. It looks like we already have a few submitted. So Sonya, I'm going to be reading them to you. And the very first question is, what would be a proposed mechanism for the lower affinity antibodies producing better agonism?
Yeah, so basically for receptor clustering, one antibody needs to bind two receptors at the same time for a sort of short period of time and then repeat this process continually. So the antibody needs to bind for long enough to enable the receptor to initiate signaling, but for a short enough time to allow it to engage with further receptors. So if you have an excess of high affinity antibodies, they'll be too stably bound to the receptors to allow this kind of receptor clustering.
All right, wonderful. The next question we have from Lee is, when the affinity of an antibody is decreased to improve clustering, is it likely that specificity decreases as well?
No, the antibody itself is still specific for that particular epitope. It's just the affinity is less, so it spends less time bound to the epitope.
Okay, great. Next question. Any ideas as to why there are such differences between IgG1 and IgG2 isotypes?
Yeah, so Ben has been looking into this, and the Antibody and Vaccine Group does a lot of work looking into how different isotypes of the antibody can affect binding and agonism. And he did find that the IgG human 2 isotype antibody can self-associate, whilst the IgG1 doesn't. So there could be a mechanism where this self-association of the IgG2 kind of allows this additional receptor clustering, and so it could nucleate from where the human 2 antibodies self-associate. But there's lots of differences between the human 1 and human 2 isotypes in their flexibility and things like that. So there's many reasons that could be between the differences.
Okay, great. Next question. Why does 341G2 H2 produce rod-like structures?
Yeah, well, we don't really know. We know that 341G2 is unusual. Its epitope spans 2 of the 4 cysteine-rich domains of the CD40 molecule, whereas all the other antibodies are confined to 1 of the cysteine-rich domains. And we know that there are a lot of differences in the ways that the receptor clustering manifests itself with different antibodies. For example, we have the cell-to-cell clusters with the low affinity Chilob74 antibody, which these cell-to-cell clusters aren't evident with 341G2 itself or CP. So there are lots of differences, and we don't really know why the 341G2 causes these rod-like structures, but it's the only antibody even from the other TNFR antibodies that we've tested that shows this kind of structure.
Very interesting. All right, next question. What is the significance of the reverse correlation between area and density?
Yeah, so we think that the smaller, denser clusters seem to have an advantage in terms of activation, possibly because just the presence of more receptors in a confined space allows the antibody to consecutively engage with many receptors, and that will amplify the activation signal. It'll also allow more intracellular signaling, so more of the intracellular signaling molecules in one place to lead to more potent receptor signaling.
Okay, great. We had one person ask if this was going to be recorded. This is recorded. And then one more question in here. Is it possible to do 3D storm on the nanoimager? And how do you propose to evaluate the numbers of receptors?
Okay, well, yep, in principle, 3D storm is possible with the nanoimager, but it's going to take some optimization. Calculating the actual numbers of receptors is also not trivial, so we'll be doing this in collaboration with the guys at ONI. So I don't know if any of you guys want to answer that question.
Okay, we'll probably come back to that one then. And it looks like we are at the end of our questions, which is great. Okay. And it seems like we are at the end. So a very big thank you for the presentation again, Sonya. If you do have any additional questions for Sonya, please just feel free to email anyone from ONI and we can forward them directly to Sonya for any follow-up needed. And once again, this webinar was recorded. Therefore, if you need to refer back to it, you can. And thank you to all the attendees for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out that survey to provide us feedback on your experience today, or tell us if you are in need of being contacted by ONI for any information on our offerings. And thank you again, and we hope that everyone enjoys the rest of their day.
Thank you.